Hello, everyone. Sorry I could not be there in person today. If I'm being honest, my anxiety to be in front of the class is unfortunately really bad. So I've decided to stay home, especially given the weather. The roads are quite slippery from my house to the school. So here I am. My title of the presentation today is New Neurons, New Brain. This is the article I've selected for today's seminar. This research article was published in 2015 by Kim et al. And is titled GDF15 secreted from human umbilical cord blood mesenchymal stem cells delivered through the cerebral spinal fluid promotes hippocampal neurogenesis and synaptic activity in an Alzheimer's disease model. I picked this article for two reasons. Alzheimer's disease runs in my family, and we have lost many of them to this disease. Secondly, I really like the brain. It is a complex, fascinating organ that I definitely desire to learn more about. So the idea of mesenchymal stem cells and hippocampal neurogenesis in Alzheimer's disease was definitely very intriguing to me. I would like to start off with understanding Alzheimer's disease. So what exactly is Alzheimer's disease? There are many forms of dementia, each with unique symptoms caused by microscopic changes in the brain. Alzheimer's disease specifically is an irreversible degenerative brain disorder that is characterized by changes in the brain that lead to a buildup of deposits of proteins and in and around the brain. Symptoms of Alzheimer's disease include memory problems that get worse as the disease progresses, issues with thinking and reasoning, a decline in the ability to make sensible decisions and judgments, performing familiar tasks and activities become more and more difficult as the disease progresses, changes in personality and mood, which include depression, mood swings, wandering, delusions, and aggression. Risk factors. Aging dramatically increases the likelihood of developing Alzheimer's disease. First symptoms typically appear in the mid-60s unless it's early onset, which in that case, symptoms would appear in your 40s, 50s. Abnormal genes can lead to a higher risk of Alzheimer's disease. There are 3 rare single gene variants known to cause Alzheimer's disease, which are amyloid precursor protein, or APP, presenilin 1 on chromosome 14, presenilin 2 on chromosome 1. And APP, the mutation would be found on chromosome 21. If a family member has Alzheimer's disease, offspring are definitely at higher risk of a diagnosis. Mild cognitive impairment also increases the risk. Bumps on the head, lack of exercise, smoking, excessive alcohol use can all lead to changes in the brain that are favorable to Alzheimer's disease. And heart health. Diseases like obesity or high blood pressure, high cholesterol are all associated with developing Alzheimer's disease. There are 2 key players in Alzheimer's disease, tau tangles and amyloid beta plaques, both of which I'm sure everyone has heard of. Tau tangles stick together to form tangles in the brain cells that block the neuron's transport system, which harms synaptic communication between neurons. And amyloid beta is a protein that forms plaques around brain cells, causing an interruption in cell-to-cell communication. So here we have a healthy brain and a healthy neuron. Here you can see the considerable size reduction in the brain in an Alzheimer's disease brain. You have your amyloid beta plaque buildups here. You've got your diseased neuron with the tau tangles within. In general, Alzheimer's disease first affects the hippocampus and the entorhinal cortex, as more neurons die off, other parts of the brain become affected. The hippocampus is a region in the brain that has major roles in memory and learning. And the entorhinal cortex is part of the brain that is basically the gateway for information entering and leaving the hippocampal area. So what exactly happens during neurogenesis? For a very long time, we were always told that each of us was given a certain number of brain cells, whether it be 100 billion or however many brain cells you were lucky to have. And that was pretty much it. But now this idea that adults don't grow new brain cells is now fully demonstrated to be completely wrong. We possess the ability to regrow brain cells through a process called neurogenesis. So that's exactly what neurogenesis is. It's the process by which new neurons are formed in the brain. Under physiological conditions, adult neurogenesis is known to occur in two regions in the mammalian brain: the subgranular zone of the dentate gyrus in the hippocampus, which is the region of the brain involved in regulating learning and memory, and the subventricular zone of the lateral ventricle, which is where new neurons are generated and then migrate to the olfactory bulb. Adult neurogenesis can be divided into 4 stages: precursor cell, early survival, post-mitotic maturation, and finally the late survival stages. I will briefly describe the process of these 4, but I won't get too much into it. But basically, you have these radial-like glial stem cells that maintain their individual pool via self-renewal, which give rise to neuronal progenitor cells, which is type 2. These 2 types of cells show similar markers, but different morphology, which then this type 2 neural progenitor cell undergoes proliferation and begins to express specific markers. So these type 2 neural progenitor cells generate neuroblasts and have more specific markers. These type 3 neuroblast cells enter the early survival stage and post-mitotic maturation stage, and basically become immature neurons, which enter the late survival stage, and immature neurons form functional connections and matured morphology and become mature neurons. There are 5 groups of molecular players that play critical roles in adult neurogenesis. So you have morphogens, which are extracellular signaling molecules. In neurogenesis, a number of morphogens were found to be critical in establishing and regulating stem cell niche, including Notch and Wit signaling pathways, which we had learned earlier in the semester. Growth factors are extracellular peptides that function as stimulants during tissue growth and development. Growth factors are known to be critical for various reasons in adult neurogenesis. And there are I listed a few growth factors here. So we have brain-derived neurotrophic factor, insulin-like growth factor 1, and fibroblast growth factor 2. Neurotransmitters obviously are involved in adult neurogenesis, including gamma aminobutyric acid, or GABA, dopamine, glutamate, and serotonin. A large number of transcription factors are found to regulate neurogenesis. To name a few here, we have sex-determining region YBOX2 or SOX2, prosporo homebox 1 or PROX1, and neurodifferentiation or NeuroD. Epigenetic factors are molecules that modify gene expression via mechanisms such as DNA methylation and histone modification. They're termed epigenetic because these modifications are heritable, but they do not involve genetic mutations. Various epigenetic factors are known to regulate neurogenesis, including methyl CpG binding domain protein 1, mixed lineage leukemia 1, polycomb complex protein BMI1, and various microRNAs. So my preceding article timeline As I said before, it was assumed for a very long time that the number of brain cells you were born with was all that you get. While you can certainly lose brain cells, you couldn't get any more. Well, in 1965, a scientist named Joseph Altman proposed to have evidence on the contrary, neurogenesis. His studies presented the first indication of the existence of newly formed dentate granule cells in a postnatal hippocampus of rats. In 1967, he proposed evidence of postnatal neurogenesis in guinea pig models. Kaplan and Bell, 1984, provided evidence of neurogenesis in the hippocampus of adult mice. Goldenall, 1999, reported neurogenesis in the hippocampi of adult primates. Fanslow and Dong, 2010, reported hippocampal neurogenesis is shown to contribute to learning and memory. Mo and al., 2011, showed evidence of altered neurogenesis in neurodegenerative diseases such as Alzheimer's disease. Kim and al., 2012, same author as my current article, showed that soluble intracellular adhesion molecule 1 reduced amyloid beta plaque buildup. He, again in 2012, showed that umbilical cord mesenchymal stem cells protect amyloid beta 42 neurotoxicity via paracrine factor. Lee et al., 2012, showed that human umbilical cord blood mesenchymal stem cells improve neuropathology and cognitive impairment in an Alzheimer's disease mouse model. And then we have my article today that GDF15 secreted from human umbilical cord mesenchymal stem cells promotes hippocampal neurogenesis in adult Alzheimer's disease mouse models. Okay. So now that I've gone through background information and my mini talk on Alzheimer's disease and neurogenesis, I'm going to dive into my article. So regenerative medicine approaches involving stem cells have long appeared to be implausible for the treatment of Alzheimer's disease. However, an alternative approach has been emerging involving paracrine factors produced by stem cells that could facilitate tissue regeneration and neuroprotection. Mesenchymal stem cells specifically secrete proteins that promote neurogenesis and neuroprotection. These findings are consistent with these authors' previous studies as mentioned in preceding articles. I have one in green here because I didn't mention it in the last slide, but both galectin 3 and soluble intracellular adhesion molecule 1 are released from umbilical cord stem cells that act in amyloid removal and as anti-apoptotic effectors. In addition, this last box here, they conducted a transplantation of human umbilical cord stem cells into an Alzheimer's disease mouse model, which resulted in an improvement in memory loss. So, the study objective is to determine if repeat cisterna magna injections of GDF15, which is a human umbilical cord blood mesenchymal stem cell secreted paracrine factor, delivered through the cerebral spinal fluid, enhances adult hippocampal neurogenesis and synaptic activity in an Alzheimer's disease mouse model. Methods and materials. All right, so the isolation and culturing of cells have been described in previous studies. But in these previous studies, they took umbilical cord blood, was collected from umbilical veins after neonatal delivery, and the mesenchymal stem cells were isolated by separation of mononuclear cells. The mouse neural stem cells were cultured according to previous studies, which I unfortunately couldn't find. The one that I did find wasn't very clear on their methods. But all these cells were provided by Dr. Y.S. Yang. The human umbilical cord mesenchymal stem cells were tested for surface antigen expression, mesodermal differentiation, and proliferation rate. Cell pellets were obtained after centrifugation and dissociated by gentle pipetting with neurobasal medium containing B27 and to supplement L-glutamine, penicillin, streptomycin, epidermal growth factor, and fibroblast growth factor. The human umbilical cord mesenchymal stem cells and the mouse neural stem cells were co-cultured in a transwalled chamber with the human mesenchymal stem cells on the top and the neural stem cells in the lower chamber. The figure here, we see the transwalled chamber, which is a culture insert used to study migratory responses of cells and consists of two compartments separated by a permeable porous membrane. The upper and lower compartments are used to culture different types of cells, and the membrane, the microporous membrane, allows for exchange of molecules. The size of pores in the porous membrane controls the passage of different molecules, and in this study specifically, the pore size was set to 1 micrometer. They then used trypan blue exclusion test to measure cell viability. This is a qualitative test based on the principle that all live cells possess intact cell membranes that exclude certain dyes, whereas dead cells do not. So in this test, a cell is mixed with a dye and then visually examined to determine whether cells take up or exclude the dye. For generation of green fluorescent protein, or GFP, mesenchymal stem cells, 3 virus particles were used to transduce the human umbilical cord stem cells containing GFP reporter gene, which is a quantitative reporter of gene expression in individual cells. In the first part of the experiment, the mice were placed under anesthesia and into a stereotaxic apparatus, which is right here, which allows for precise location of brain sections, and they were placed into the apparatus for transplantation. First, parenchymal injection, recombinant GDF15 was injected into the bilateral dentate gyri, and human umbilical cord mesenchymal stem cells were stereotactically injected into the hippocampal area. Following the first procedure, the mice were given an intraperitoneal injections of BRDU twice a day for 24 hours, plus intraparenchymal administration of GDF15. In the second part of the experiment, the mice were placed under anesthesia again placed into the stereotaxic apparatus in a position that allowed for the cistern omega to be at the highest point and 15 microliters of human umbilical cord stem cells were injected. For quantification of the human umbilical cord mesenchymal stem cells remaining in the brain tissues after both injections, homozygous athmic rats were used, which these rats are hairless, lack normal thymus gland, and have a defective immune system because of a genetic mutation. Growth medium was then collected from the neural stem cells, the human mesenchymal stem cells, as well as co-cultures of both cell types. To detect the secreted proteins, the human cytokine antibody array 9 was conducted according to manufacturer protocols. A human cytokine antibody array 9 is an antibody pair-based assay type which allows for the detection and quantification of cytokines in a biological sample. Membranes were incubated with a blocking buffer at room temperature, then at 4 degrees overnight. After washing, the membranes were incubated and diluted with biotin conjugated anti-cytokine antibodies for 2 hours at room temperature. Then after another wash, the membranes were then incubated in horseradish peroxidase conjugated streptavidin for 2 hours at room temperature. The signals were detected with ChemiDoc system, which is an imaging system, and quantified with ImageJ software, which is a public image processing software. siRNA, if you recall, is small interfering RNA, which is able to regulate the expression of genes. The human umbilical cord mesenchymal stem cells were treated with GDF15 siRNA for 6 hours. Then these treated cells were cultured in a complete media overnight and co-cultured with mouse neural stem cells. Control siRNA with at least 4 mismatches to any human, mouse, or rat genes were used for this study. Total RNA from cells was obtained with trizol, which is a newer developed reagent used in the extraction and deproteinization of RNA. Complementary DNA, or cDNA, was synthesized from the total RNA with superscript 2 polymerase, which is an enzyme used to synthesize cDNA from RNA templates, and oligo-DT primers, which prime the reverse transcription reaction. So priming with this type of primer initiates the synthesis at the 3' end of the RNA fragment. PCR was performed with gene-specific primers. The list of primers you see here are just 4 gene-specific primers used for quantitative RT-PCR analysis. Forward primers attached to the start codon of the template DNA, while reverse primers attached to the stop codon of the complementary strand of DNA. The products of PCR were run for semi-quantitative gene expression analysis. The study generated a standard curve for quantitative analysis of real-time PCR using a mixture of athmic rat CSF and various quantities of the human umbilical cord mesenchymal stem cells. DNA extraction was performed using DZ blood and tissue kit. Examination of human-specific polymerase 2, peptide A, or polar 2, were both forward and reverse, with both forward and reverse primers. The PCR cycle consisted of 3 initial steps: denaturation, amplification, and the final elongation. Denaturation for 10 minutes, amplification for 45 cycles, and lastly, 30 seconds of elongation. Immunoblot analysis was performed, which is a technique that uses antibodies to detect specific proteins in a sample. Each membrane was incubated with various antibodies, including anti-SRY box2, anti-nestin, anti-beta-actin, or anti-amyloid beta 42, to name a few. Immunohistochemistry, or IHC, was performed on the brain tissues of euthanized mice, which is a method that uses antibodies to check for certain antigens/markers in a sample of tissue. They use various antibodies, including anti-SOX2, anti-AB protein, anti-GDF, and anti-microtubule-associated protein 2, or MAP2. All images were acquired using a confocal microscope and a light microscope. The hippocampal regions were dissected from 1-day rats, dissociated and plated onto a coated cover slip for 14 to 21 days. Constructs were then transfected 8 days after plating was completed, And then imaging was performed 14 to 21 days after the plating with a laser illuminated epifluorescence microscope and analyzed with ImageJ. Finally, the results. So I did find the supplementary material, and I have decided to include all of it into my presentation for more information on what the study covered, but I will only briefly go over this material. So here we have the supplementary figure 1, which is when they first quantitatively analyzed human umbilical cord mesenchymal stem cells that remained in the brain tissues after parenchymal or cisternal magna injections with quantitative PCR. So figure 1A, we have the number of human umbilical cord mesenchymal stem cells that remained in the brain after single injection into the brain parenchyma of the hippocampus. You can see here that the human umbilical cord stem cells lived no longer than 8 weeks after a single injection into the parenchyma. In figure B, we have a number of mesenchymal stem cells that remained in the brain after 3 repeat injections through the cisterna magna delivered through the CSF. The cells were not completely vanished after the 8-week mark and after the 3rd injection. In figure C, we then here see that the number of human umbilical cord mesenchymal stem cells that remained in the tissue after 3 injections was 3.55-fold higher than that after a single injection in the parenchyma. All right. This supplementary material on the previous slide suggested that repeat administration was optimal for the purpose of the study. Their next step was to perform an immunohistochemistry of green fluorescent protein labeled mesenchymal stem cells within the brain parenchyma. After single and repeat injections of GFP labels mesenchymal stem cells through the cisterna magna, the distribution of these cells was determined by detecting green fluorescent protein fluorescent signal. These GFP positive human umbilical cord mesenchymal stem cells were observed in all regions of the brain parenchyma. So we have single injection 4 weeks and single injection 10 weeks, where here we see a comparison between the weeks after injection. You can see from week 4 to week 10, you can see that almost no living human umbilical cord mesenchymal stem cells were noticed in week 10. There's compared to all the green in these photos, compared to all the green in these photos, a lot of it died off. Whereas looking at repeat injection after 10 weeks, the cells are still alive at 10 weeks and are very comparable to the results they got at single injection at 4 weeks, except even better. The third step was to look at the effects of single versus repeat injections on the expression of nepricillin, or NEP, which is an amyloid beta degrading enzyme, and amyloid beta clearance. In the previous study mentioned here by Kim et al. 2012, the study mentioned they demonstrated that a single injection of mesenchymal stem cells into the parenchyma does reduce the levels of soluble and insoluble amyloid beta through the overexpression of NEP. Therefore, they wanted to determine whether this effect could be replicated by human umbilical cord mesenchymal stem cell delivery through the CSF. So they measured NEP and amyloid beta levels. As they expected, between repeat and single injections, the NEP levels increased and amyloid beta levels decreased by 8.3% in single injection and 50.8% in repeat injections. These results were validated with IHC. So this is another supplementary figure. These results where you can see here that the diminished AD plaques were detected in the dentate gyrus, which I will refer to as GD or DG from now on, and the thalamus. So effects of human umbilical cord mesenchymal stem cells in the adult hippocampal neurogenesis. Because CSF administered human umbilical cord stem cells were found in two neurogenic regions, the SVZ, which is the subventricular zone, and the DG. They wondered if there may be a role of human umbilical cord mesenchymal stem cells in regulation of adult neurogenesis, which could restore cognitive function in Alzheimer's disease mice. So they measured the level of neural stem cell proliferation and differentiation. Both single and repeat injections showed increased levels of adult neural stem cell proliferation and differentiation markers. To confirm these results were due to either an increased expression of the marker or an increased neural stem cell population, they did stain for the markers. The following followed by they first stained for neural stem cell markers, which included SOX2 and glial fibrillary acidic protein, or GFAP, and the DG. Followed by staining mature neuron markers, which in this case included neuronal nuclei, or NU, in the DG as well. A quantitative immunohistochemistry analysis indicated that in both single and repeat injections of human umbilical cord mesenchymal stem cells, neural stem cells and mature neuron populations in the DG increased based on single and repeat. Both neural stem cell and mature neuron populations in the DG increased, indicating that both injections enhanced hippocampal neural stem cell populations. In the supplementary material provided, they also measured neural stem cell and neuroblast population in the subventricular zone. After a single and repeat injection of mesenchymal stem cells, the number of neural stem cells increased. So you can tell here, after single injection, and then much more mesenchymal stem cells were noticed after repeat injections in both SOX2 for neural stem cells and neuroblast populations. Yes, so number neuroblast populations increased as well. These results were validated by administration of HS68, which are human fibroblast cells, into the CSF, and showed that there was no significant changes between the subventricular zone and the dentate gyrus. Therefore, these results show that CSF-administered human umbilical cord cells promote neural stem cell proliferation and differentiation in both the subventricular zone and the DG, and that repeat administration enhances these effects. Identification of GDF15 as a human umbilical cord mesenchymal stem cell secreted paracrine factor for stimulation of adult neural stem cell proliferation in vitro. All right, so the effects of human umbilical cord mesenchymal stem cells on adult neural stem cell proliferation were evaluated in vitro. This was done by co-culturing human umbilical cord mesenchymal stem cells with adult neurostem cells in the transwell chamber, as discussed in the materials and methods section, and used trypan blue exclusion tests for quantifying living and dead neurostem cells. The number of live neurostem cells increased in a time-dependent manner when they were co-cultured with the human umbilical cord mesenchymal stem cells, as you see in figure A. So as time went on, more co-culture cells were living. In supplementary figure 3A shown here, after co-culturing, the cell viability of neurostem cells in the lower chamber was measured, and this graph is showing the number of viable neurostem cells in a percentage after 1 to 3 days. So as you can see, the percentage of cell viability increases in a time-dependent manner. Furthermore, shown here, the levels of adult neurostem cell proliferation markers were significantly upregulated in neurostem cells, which suggests that human umbilical cord mesenchymal stem cells do stimulate neural stem cell proliferation through the secretion of soluble factors. To identify soluble factors secreted from human umbilical cord mesenchymal stem cells, cell media co-cultured was analyzed. In figure 3, you have neural stem cells and human umbilical cord mesenchymal stem cells alone, then the co-culture media. The levels of GDF and plasmogen activator inhibitor 1, or PAI1, were increased in the co-culture compared to the controls. Sharing neural stem cells and your mesenchymal stem cells alone, and then the co-culture media. The suppression of GDF15 expression in human mesenchymal stem cells as a result of GDF15 siRNA resulted in the decreased levels of expression of SOX2 and nestin in neurostem cell co-cultures. They then reversed the effect of the addition of recombinant GDF15 by the addition of recombinant GDF15 in the co-cultures, which resulted in the upregulation of nestin, SOX2, and PNC expression due to the presence of the recombinant GDF15. GDF15 positive mesenchymal stem cells were also observed in the DG and the SVZ of Alzheimer's disease models, as shown here in figure 3E. The role of GDF15 in the stimulation of adult neurostem cell proliferation in vitro and enhancement of mature neuron population in the DG. To better understand the importance of GDF15 functions involved in hippocampal neurogenesis and neural development, they examined whether hippocampal neurogenesis was regulated by recombinant GDF15 in a concentration or time-dependent manner. Neural stem cell markers from adult neural stem cell post-treatment with either 1, 2, or 100 ng per milliliter of recombinant GDF15 was administered to mouse adult neural stem cells for 24 hours, as you see here in figure 4A. The results of this showed that nestin, SOX2, and pCNA markers increased after the treatment of 20 ng per milliliter of GDF15, making this the optimal concentration. Now that the optimal concentration was determined, neural stem cell markers as a function of treatment duration was analyzed, as you see here in figure 4b. In figure 4b, they administered 20 ng per milliliter of recombinant GDF15 to adult neural stem cells for 6, 12, and 24 hours. You can see here and here that the levels of neural stem cell markers gradually increased in response to the duration of the treatment. To understand the effects of GDF15 in adult hippocampal neurogenesis in vivo, varying concentrations of recombinant GDF15 was administered directly into both hippocampi, and after 24 hours, the DG sections were stained with antibodies for neural stem cells, so SOX2 and GFAP, and mature neurons, which was NeuroN. Figure C is showing the presence of SOX2 and GFAP double positive cells. Do you have your... So after 24 hours of administration of 0.5 micrograms per kilogram of GDF15, the number of adult neural stem cells was significantly increased in response to GDF15, almost 1.5-fold more. After 24 hours of administration of 1 microgram per kilogram, GDF15 resulted in a 36% decrease of neural stem cell population. The number of mature neurons in the DG was increased in response to GDF15 in a dose-dependent manner, as seen in figure 4D. This figure 4D is showing the presence of NU positive cells in the DG at 0.5 and 1. Based on the above results, they further examined whether GDF15 would lead to proliferation of adult hippocampal neural stem cells and differentiation into neurons within a 24-hour period. So figure 4E is just showing a schematic of recombinant GDF15 and BRDU injections, which were every 12 hours, into the cisterna magna and of mice, and their scheduled animal sacrifice. After 24 hours post-injection of GDF15 and BRDU, the DG sections were stained, and BRDU and NU positive cells in the hippocampi were quantified. The number of positive cells in the GDF15 administered brains increased by 3.82-fold, suggesting that GDF15 stimulated neural stem cell proliferation and differentiation into neurons within the studied 24 hours. You have a huge increase in mesenchymal stem cells and mature neurostem cells based on their control group. Therefore, it was determined that GDF15 to be a causative factor that enhances the expression of both neural stem cells and SOX2 and nestin, as well as cell proliferation markers, which was PCNA in vitro. It was also determined that GDF15 enhances proliferation and differentiation of neural stem cells in the adult DG in vivo. Efficacy of human umbilical cord mesenchymal stem cells in the enhancement of synaptic activity in vivo and the potentiation of adult hippocampal neurotransmission through action potential stimulation in GDF15 in vitro. To determine whether human umbilical cord mesenchymal stem cells can induce synaptic activity, they analyze the expression of synaptic vessel markers, including synaptophysin, or SYP, vesicle-associated membrane protein, or VAMP, syntaxin, syntaxin, or STX, and synaptogen, or SYT. I'm sorry if my pronunciation of the words are wrong. I'm not very good. But anyway, by injecting all those synaptic vesicle markers by injecting human umbilical cord mesenchymal stem cells into the CSF of adult Alzheimer's disease mice, in figure 5A, we have a single and repeat administration of human umbilical cord mesenchymal stem cells, which were extracted and analyzed. After injections, the level of all synaptic vesicle markers had increased within the entire brain. In an IHS and histological analysis, the brains were sectioned and stained with anti-MAP2, which is in green, and anti-SYP, which is in red. The levels of SYP in mature DG increased post-repeat injection of human umbilical cord stem cells, which confirmed that they do enhance SYP expression. After obtaining the evidence that human umbilical cord mesenchymal stem cells increase expression of synaptic vesicle markers, the neurotransmission of hippocampal neurons with action potential stimulation was measured after co-culturing human umbilical cord mesenchymal stem cells and hippocampal primary neurons that were transfected with a conjugated vesicular glutamate transporter 1, or VGPH, for 24 hours. Based on the results, synaptic transmission was detected in primary neurons when VGPH responses were monitored after 100 action potential at 10 Hz, then exocytosis of synaptic vesicles was increased. So to show this in figure 5C, this is showing VGPH images at rest and the difference in images at 100 action potential and during NH4CL validation procedure. So the validation procedure. To validate the above results, they applied Na4CL to the neurons. In the supplementary figure 4B, you see a representative average trace of exocytosis from control, which is in black, and the mesenchymal stem cell co-culture neurons, which are in red. As you can see, the signals you see between the two groups did not differ significantly at all. Since paracrine factors that are secreted from human umbilical cord mesenchymal stem cells potentiate synaptic transmission, they next examined the possible role of GDF15 in direct potentiation of activity-driven synaptic activity. They did this by using pH transfected primary neurons that were stimulated at the 100 action potential at 10 Hz for 10, 20, 30, or 60 minutes after the addition or the absence of GDF15. In the supplementary 4C, we see a time course trace of synaptic vesicles exocytosis from control, which is in black, and GDF-treated neurons, which you see in red. As you can see, the longer the neurons were stimulated with the presence of GDF15, the response grew and increased, as you see here. You can also see in figure 5D, the application of GDF15 resulted in a time-dependent potentiation of synaptic transmission. As time went on, based on the control and the GDF15, the synaptic vesicle release increased. In summary, the paracrine action of human umbilical cord mesenchymal stem cells did in fact promote both hippocampal neurogenesis and synaptic activity in hippocampal neurons through GDF15. Finally, discussion. I'm really sorry if the results were kind of all over the place. I'm not that great at oral presentations, so I hope it was okay. But in this study, they demonstrated that repeat intra-athecal delivery of human umbilical cord mesenchymal stem cells could have therapeutic effects on Alzheimer's disease pathology and promote hippocampal neural stem cell proliferation, neural population maturation, and synaptic activity in Alzheimer's disease mice through GDF15 paracrine factor. So two major implications arose from this study. The first being that intrathecal administration of these cells promote hippocampal neurogenesis and synaptogenesis. The study demonstrated that adult hippocampal neurogenesis was promoted through human umbilical cord mesenchymal stem cell paracrine factor GDF15. The study showed the suppression of GDF15 synthesis resulted in the absence of stimulation of neural stem cell proliferation, whereas the addition of GDF15 promoted in vitro and in vivo proliferation of neural stem cells and the in vivo maturation of neuron populations. Besides the enhancement of hippocampal neurogenesis, synaptic activity of hippocampal neurons was also stimulated in Alzheimer's disease mice due to the secretion of GDF15. In this study, they were not able to determine the exact underlying mechanisms of how GDF15 potentiates synaptic activity in a short period, but they did come up with three possibilities. One is that GDF15 might be able to activate voltage-gated CA2 channels, resulting in an increase of CA2 plus influx at the presynaptic terminal. Two, GDF15 might be able to redistribute voltage-gated CA2 plus channels at the release site. And three, GDF15 might be able to affect the assembly of the synaptic vesicle fusion machinery that increases synaptic vesicle release. Major implication 2. The second major implication for future clinical trials is that these administration could be more advantageous delivery route for repeat delivery of human mesenchymal stem cells into the brain rather than a direct transplantation into the brain kymigma. This leads me to a clinical trial. So in 2015, right before this article was done, a clinical trial titled Stereotactic Brain Injection of Human Umbilical Cord Mesenchymal Stem Cells in Patients with Alzheimer's Dementia was done by direct transplantation of human umbilical cord stem cells directly into the brain kymigma. This trial had numerous obstructions from a limited lifespan of the cells, technical limitations, and high level of invasiveness from brain surgery, essentially. This led for a less invasive and more feasible route for repeat administration. So they came up with the delivery of cells through the cerebrospinal fluid to the cisterna magna. They came to the conclusion that CSF, because human umbilical cord stem cells were more likely to disperse to the lateral ventricle and begin migrating to the parenchyma, including the hypothalamus, the cortex, and the two areas in which neurogenesis occurs, CVD, or the CV, the SVZ, and the DG. Results of the clinical trial, no patients did show serious adverse effects during the 24-month follow-up period. During the 12-week follow-up period, there were common complaints, which included like headache, dizziness, pain from the procedure. Therefore, the administration of the human umbilical cord cells was concluded to be safe and feasible, although it was just really highly invasive. In conclusion, this study found that the efficacy of repeatedly administered human umbilical cord mesenchymal stem cells through the CSF significantly influences the removal of toxic amyloid plaques in hippocampal neurogenesis and synaptic activity in mature neurons through GDF15 in Alzheimer's disease mice. Follow-up articles I'll just go through real quick. I can't pronounce his name, but in 2017, evidence that human umbilical cord stem cell xenografts improve cognitive decline and reduce amyloid burden in a mouse model of AD. 2017 again, through the same author that did this study that I just reviewed, titled where he looked at thrombospondin 1, which is secreted from human umbilical cord cells, which rescue neurons from synaptic dysfunction. Kim and al. Again, same author in 2018, provided evidence that GDF15 positively affects amyloid beta levels in vitro and in vivo of Alzheimer's disease mice. And Lim and al. 2020, evidence that galectin-3 secreted from human umbilical cord mesenchymal stem cells reduces tau phosphorylation in an Alzheimer's disease model. Zhang et al. 2022 suggested that mesenchymal stem cells are a multi-target cell therapy for clearing amyloid beta in Alzheimer's disease. And then I can't pronounce her name either, but in 2023, the most recent article I could find provided evidence that the expression pattern of GDF15 in the human brain does change during aging in Alzheimer's disease. Critiques. I don't have too many critiques for the article. Unfortunately, like all biology students, never got a course where we could critically review articles for the whole semester, so I'm not the greatest at it. But a couple negatives that I did come across, that I was a little confused throughout the article when it came to their test models. They mentioned a few different mouse models, including APP-PS1 mice, Sprague's Dolly mice, transgenic mice, double transgenic mice. This is very likely could have been just my lack of clarity, but I was just a little bit confused as to what their main AD mouse model was. Previously described in. By this I mean very often throughout the article, they would refer to previous studies when talking about previous results, or when describing previous methods used in another study. For example, in the materials and methods section for cell preparation, the very first sentence was, and I quote, "The isolation and culturing of human umbilical cord mesenchymal stem cells have been described in previous reports." I understand why they do this. I just don't like it. I like having to go through different articles, trying to find what they did and how they did it, but it was very time consuming. So I didn't overly like that. Another dislike was that there were limited statistics. They had many facts supported by imaging and graphs, which is great, but all through the article, they would say this was significantly upregulated by 1.58-fold, for example. They would provide the new value that they proved, but no previous statistic for comparison. Statistics were mainly shown in figure captions. I actually haven't really seen this before. I didn't like how they only included the statistics in the figure captions. I found this to be a little complicated with having to constantly flip back and forth between pages. I think all significant statistics should be reported in the paragraph that they are being discussed. Things I like, other than the complaints above, the article did generally have good flow despite having to flip back and forth for statistical values. I liked how each section was labeled with the appropriate and descriptive subheadings. Figures are clearly labeled and easy to reference. Validation procedures and supplementary materials were referenced throughout. I did like this, and I enjoyed that all the validation procedures were briefly described in the paragraph that they are associated with. And all reference material was referenced throughout and easy to find. They had a PBS control group, which I liked. This allows for comparisons between interventions that were clear to read. These are some references. All the other references were in my preceding follow-up articles, so they were in text references. And that's it. I do believe Dr. Nickerson will be posting this on Blackboard. Please ask questions. There will be a discussion board, I believe, posted for me to answer your questions. So please feel free to ask away. I will keep an eye on it and get back to you when I see it. Thanks, everybody.